Okay, we are just recording the talks. So thank you for coming to this second webinar of the French Purine Club. Happy to welcome you. So we will have today 3 speakers following the order on the announcement. So we will starting by Alexander, followed by Maria and Ken to conclude. We will ask the question, can ask the question at the end of the talks, either by raising your hands or by putting your question in the chat if you prefer. So we can start with Alexander. Alexander, if you want to put your presentation on the screen and make a brief introduction. So Alexander Pfeifer is directing a lab in the Institute of Pharmacology and Toxicology at the University of Bonn. He's mainly working on studying the molecular pharmacology of the adipocyte tissue in the context of metabolism. Is working of many signaling pathway, particularly purinergic signaling. He will be here today to talk mainly about this aspect. So Alexander, the Zoom is yours.
Merci beaucoup, and thanks a lot for the introduction and the kind invitation. As I just said before, I think it's a very nice idea or a very nice thing to have these purine meetings and focusing on purinergic signaling. So as you just said, Eric, we are focusing, I didn't dare to say purines in fat because no one likes fat or adipose tissue, and a lot of people think it's boring, but I hope to convince you that it has some interesting aspects. So I'm here at the University of Bonn, and we have a collaborative research center funded by our German funding agency, and it's called But Energy. It's about adipose tissue. So why we are interested in adipose tissue? It's because of the obesity pandemic. As most of you know, hundreds of millions of adults are obese, and it's not only affecting the rich countries, so to say, France, USA, United States of America, the European Union, but also it's a worldwide problem, as you can see here, also in Northern Africa, and the obesity prevalence doubled within this short time period. And there's a 100% increase in the number of people affected. And as you can see here in this color-coded map from the WHO, it's a worldwide problem. And for example, in China, although the numbers were still very low at that time point, meanwhile, there are more than 120 million diabetic people living alone in the People's Republic of China. So that is more than the German population together with the Belgium and the Netherlands, I think. So overall, overweight and obesity are linked to more death worldwide than underweight, which is, I think, a very frustrating observation. And so we and other labs are very interested in adipose tissue. I mean, many labs are looking into the central nervous system and regulation of appetite, which for sure is very, very important. And these new GLP-1 inhibitors, Ozempic, for example, are very effective. We are interested in the fat because fat is the main energy storage, and obesity is defined as the increase, the abnormal increase in white fat tissue. So on the other hand, there is a completely different kind of fat, which was long time overlooked, and it's called brown fat. And it's called brown because these adipocytes contain a high number of, or have a high content, I should say, of mitochondria. They have as many mitochondria as heart muscle cells. And they are very vascularized, these tissues, these brown fat tissues, and this is why they're also brownish. And the main task of these strange adipocytes is not to store fat like the white adipocytes that normally we don't want to have, but these adipocytes are wasting energy. They're dissipating energy in the form of heat, And they have a very special protein. It's called uncoupling protein 1 that they express in their mitochondria. And this disrupts the electron gradient that normally leads into production of ATP. I don't want to go into details because we have not much time. But so to make a long story short, so these adipocytes and these adipose tissue depots, these brown fat depots, are very important for mammals and for us humans also, especially after birth and when we are very young, to keep our body temperature up, even in cold stress. And because a lot of this brown fat then degenerates and is lost, this is not very well understood. It was long believed that we adults do not have brown fat. But it was shown several years ago that indeed also human adults have active brown fat. And it was also found that in our human white fat tissue, especially in the subcutaneous fat, there is strange brown-like fat cells that are also multilocular here and contain not so much fat, and they also express the UCP1 protein, and they can also dissipate energy. So it would be important to understand how this overall thermogenic fat, it's called, whether it's brown or beige, doesn't matter now, is regulated. And so to understand also perhaps the physiological importance of this adipose tissue, I think this study here from the Netherlands is very important. And this showed that if you plot body mass index versus brown fat activity, those people that are very slim have a lot of brown fat. And the brown fat in the human adults is mainly here in the neck area and in the central body parts, and also close to the kidney. And recently there was a very interesting paper here in Nature Medicine from the Cohen lab, and that showed that brown adipose tissue is associated with cardiometabolic health. And they studied 50,000 patients. And those that had brown fat were much more healthy concerning cardiovascular disease than those without brown fat. So that's the introduction, so to say. So brown fat is normally activated by cold. And so then cold exposure induces the sympathetic nerves to release norepinephrine. And then this activates energy expenditure in these brown adipocytes. So far, so good, but we cannot use norepinephrine in the clinics. And so we set out to find, we and other labs, to find other alternative or synergistic molecules or hormones that could activate or regulate brown fat. And so we were focusing on ATP because this is known as a known co-transmitter during sympathetic nerve activation. And we are very much focusing on adenosine, and as you will hear in a minute, also on inosine, because the ATP is very rapidly degraded. So what we did to analyze this is a very old-fashioned experiment. And we thought, if we isolate brown fat from mice, so this is here, this brown bean-like, this is how it looks like. It's small in the mouse, and it looks like a bean-like structure. And we put it into an old chamber that we found in our institute. It was it's an electrical stimulation chamber, and we wanted to activate the sympathetic nerve endings in the adipose tissue. And so this is why we activated this 10 Hertz pulses to release then the remaining neurotransmitters or vesicles that contain the neurotransmitters in the tissue and in the supernatant. And so what we found is, here is the control is green here, tetrodotoxin, to block the sodium channels and the release of the neurotransmitters. What you can see is the stimulation induces here, starting at 2 minutes, a very nice release of norepinephrine here. And on the lower left, you see the release of ATP in the tissue, in the brown fat, mimicking, so to say, sympathetic nerve activation. And we also found adenosine. So whether the adenosine is now released from the nerve endings or generated by this cascade of enzymes that then produce adenosine out of ATP and AMP, that's not completely clear so far. But what we did is we analyzed knockout mice that lack CD73 and RTP release in this mice, and this brown fat from these mice is very normal. That is not affected. But when we check now the adenosine, the ATP release is not affected. But when we check now adenosine generation, so to say, or adenosine concentrations, then we see it's much lower here in green in the knockouts. And the peak here is always wild type versus knockout, wild type versus knockout. Basal is lower, the peak is much lower. And when we check here wild type, see there's a nice increase in adenosine, but in the knockouts, this increase is much, much smaller and not significant. So it looks like that CD73 plays an important role, at least in the brown fat, to generate adenosine out of the ATP. And so we are very interested in the adenosine because when we put ATP on the brown adipocytes, they don't do a lot, or they don't react to the ATP in a significant way. But so we were thinking perhaps adenosine and P1 receptors are important. And so what we did initially, we checked for sure expression of these are the P1 receptors, as you all know, A1, A2A, A2B, A3. The orange reddish ones are GI coupled, and the A2A, A2B receptor are GS coupled, and those are for sure very interesting. GS coupling would then lead to cyclic AMP, and cyclic AMP is the main activator of adipocytes, no matter whether they are brown, white, or whatever color. So, and what we found is when we check brown adipocytes, here on the left, brown adipose tissue, then here white adipose tissue, and then brain in a Western blot for A2A. And as a positive control, we transduced the brown adipocytes with a lentivirus overexpressing A2A to make sure that we really detect the right band. And then we also employed mice that we got from AGIC lab, the A2A knockout mice. And you can see here, there is a nice expression of A2A, especially in the brown fat and not so much in the white fat. So when we check now the knockout mice, and what you can see here is a thermographic analysis. This is non-invasive of brown fat thermogenesis with the infrared camera. So these are newborn mice. And if you take away the mother for a short time, then the brown fat here in this wild type pup, they don't have yet fur, starts firing. Excuse me. So the brown fat is activated by this cold stress, so to say, when the mother is not there to protect them or to give them warmth. And here you see the A2A knockout mouse, and see here white is very hot, red and orange is warm but not so hot. So it's not as hot, or it cannot increase temperature from the brown fat or in the brown fat as the wild type glutamate. And this is here the indirect calorimetry, which is the gold standard in our field for energy expenditure derived from the brown adipose tissue. And you can see here a clear, significant reduction in thermogenesis in brown fat activity. On the other hand, if we check, then if we use an A2A agonist that I got from Christa Müller, and here we have vehicle, no big change in the oxygen consumption, energy expenditure. If we give the mice norepinephrine, there's a nice increase, like cold exposure. Norepinephrine is the mediator of brown fat activation. But when we activate the A2A receptor specifically, we also see a very nice increase in energy expenditure. So this shows that the P1 receptors are important. Adenosine is produced during brown fat activation. And next we asked, is this also an autocrine paracrine factor? Sorry, I have still a cold. So can the adipocytes themselves release or generate purines, not just the sympathetic nerves, so to say? What is the source of the purinergic molecules, the adenosine, for example? Is it only ATP released from the sympathetic nerves, or is it also the cells themselves? And so, sorry. So what we did is we did a metabolomics analysis of the brown fat. And what I want to do now is I want to switch gears. I want to go away from activation of brown fat. I want to address an important question with you. And this is that BAT activity, I showed you that before, declines in obesity. So this is the big elephant in the room, so to say. People working on brown fat always say, oh, we need to activate brown fat. That's very important, and that's people will be healthy. But how can we do that in obese people that don't have brown fat, or it's not activated, or it's not active? And so this is also a picture from the Dutch group from Van Marken-Lichtenberg, and they did inject radioactive glucose into subjects. And then they did cold exposure. And the radioactive glucose is immediately taken up by the brown fat. Brown fat, once it's activated, takes up 75% of ingested glucose. And so what we see here is this brown fat is in the neck, axillary region, and here close to the heart. And when he puts, or when they put these subjects, these young, healthy men into terminal condition, it's completely switched off. And if you do that longer, if you expose a mouse for 4 weeks to 30 degrees Celsius, the brown fat completely disappears. What you see then is only something like white fat left over. And so I'm sitting here, you know, in my warm office, and when it's cold in the winter, I put on the heating and so on. So we all try to be in terminal conditions. So what does this mean for our brown fat? It means that we don't have a lot, presumably. And so we wanted to understand how this process functions, or what is going on in the brown fat. So what we did is we exposed the mice to terminal neutrality. A mouse is terminal neutral at 20, but at 22 degrees like we are, but at 30 degrees. And we checked for apoptotic cells. So because our hypothesis was that the brown fat cells don't like that, and that the brown fat, I mean, you just saw that is diminished. And we think that apoptosis plays an important role. And indeed, there's a significant increase in apoptotic cells. Also, marker genes for apoptotic cells was increased in the adipocytes from these mice. So talented graduate student in my lab, Laura Pütz, she established a model for apoptosis in brown adipocytes. She treats them with UV light or with nuclein-3, and she is very careful not to disrupt the membrane, because you would say, sure, all the purines that are inside the cell are then spilled out, but she wanted to make sure that these are intact cells, pre-apoptotic. And so we took the supernatant from these pre-apoptotic cells and put it onto healthy brown adipocytes, because we thought perhaps there are some metabolites that then regulate neighboring cells. You know, perhaps they have some influence, perhaps there are some important signals, and perhaps with these signals we understand this process of brown fat loss in obesity and in terminal neutrality better. And to our big surprise, when we treated, or when she treated these cells, these healthy cells, with the supernatant from the apoptotic or pre-apoptotic cells, she found that they increased the expression of this very important UCP1 protein. So they are doing better. So this is something positive for them. So it's like a cry for help of these dying cells to the neighboring cells. So we wanted to understand what metabolites these are. And this is a Purine meeting, the Purine Club from France. So for sure it has to do something with purines. So the untargeted metabolomics that we did took us quite some while to establish that, clearly shows that those that are upregulated, those metabolites that are upregulated in the apoptotic cells that are secreted by the apoptotic cells, these are mainly, or one of the biggest group is purines. So you see here AMP, inosine, and so on. And so therefore we went on to study these purines. So we wanted to understand which purines are the most important ones. So we found that in this cell model system, ATP is induced or secreted by these cells, but AMP is much higher. Inosine is a very important purine signal or purinergic molecule, and also for sure hypoxanthine. So the main metabolites that we find are IMP, inosine, and hypoxanthine. And next step is we wanted to understand if one of these purinergic molecules could mediate this positive effect on the healthy cells. So what we did is we put AMP, inosine, hypoxanthine on the cells and checked for cyclic AMP production, because that is the main secondary messenger that activates brown adipocytes and UCP1 expression. And only inosine was giving a significant increase in cyclic AMP. So this is why we focused on inosine. So what we did is we did multiple studies. I show you now what I think are the most important ones. So we treated mice that have already established obesity, obese mice, with inosine injections, and we checked what's happening. And so here you can see in dark the vehicle injected mice. So they are obese. I mean, they are very heavy. 45 grams mouse is a really obese mouse. And those injected with inosine, they lose weight in a significant way. And here you can see the body composition, lean mass here in gray. So here, those that were injected for 4 weeks with inosine, they have more lean mass and much less fat mass. So then inosine appears to be important for these brown adipocytes, or at least for the metabolism in these mice. And we wanted to understand how it's endogenously regulated. So it has to be generated somehow. So we think it's generated from ATP, released and then degraded, finally by the ADA. But there's also these ENTs, these equilibrative nucleoside transporters, and they transport adenosine and inosine in and out of the cells depending on the concentration gradient. So I have to hurry up now. And so we checked the ENT1. This is encoded by the gene SLAC29A. And we found that this is nicely expressed in the brown adipose tissue. And when we knock out ENT1 or in the study knockout cells, we see that the extracellular concentration of inosine is increased. So next, we wanted to use a pharmacological approach. We are pharmacologists. So we used dipyridamole, which is an ENT1 inhibitor. And here is the energy expenditure, here wild-type mice. And then those mice treated with dipyridamole here in red have a significant increase in energy expenditure, and it's not further increased in the knockout mice. So we think this is really mediated by these ENT1 channels. And interestingly, when we treat the mice, when we also expose them to cold for 7 days, we see a more increased browning of the white fat, so more thermogenic adipocytes. So, and then to wrap up and to come to the end, finally, I want to show you some data on the human situation. So we were thinking about what does ENT1, could ENT1 play a role in human metabolism? And we teamed up with a group in Leipzig, here in Germany, geneticists that have a population of 900 patients that they're very nicely characterized. And they checked for a variant of this channel, of this SLAC29A, and they found a variant which we further analyzed, and this is a loss of function variant. And those people that carry the loss of function variant, they have a lower mean body mass index. And this is shown here also in this color code. So the wild type, so to say, the population that has not this loss of function variant, the underweight here in blue is healthy weight and orange overweight. And if you look at this loss of function mutant, these people are more healthy in the healthy weight range than the people that have not this mutation. And the odds ratio of being obese is 59% compared to the wild type variant, so to say. So to wrap up this, what I showed you today, the extracellular purines play a very important role in regulation of adipocytes and adipose tissue, especially of brown fat. Adenosine and inosine can activate the P1 receptors that again then activate brown fat thermogenesis. Presently, we are also looking at the channels or transporters that transport ATP out of the cells, because we think that's also important. There was a recent paper by the Leitinger group that showed that pannexin 1 has an important role there. And this is my summary. And I want to thank you for the kind introduction and invitation and for your attention. Thanks a lot.
Thank you, Alexander, for this nice and short presentation perfect on time. So as you wrote in the discussion, we will ask the question at the end of the three talks. So don't hesitate to raise your hand at the end or to write your questions. So now we will switch to Maria Domercq and on P2X receptors. So Maria, can you share it? So Maria Domercq is a professor at the University of the Basque Country in Bilbao and working as a group leader in the Achucarro Neuroscience Center. She's mainly interested in the role of glial cells and oligodendrocytes in physiopathology, particularly in multiple sclerosis. And these last years, she focused mainly on the role of P2X receptor, P2X7, and then P2X4 receptor in microglia and oligodendrocytes and on multiple sclerosis. So, Maria, when you want.
Thank you, Rick, for this introduction and this kind invitation. So I'm going to change the topic, as Eric says, and my talk is going to be focused on multiple sclerosis. So for those who are not familiar, multiple sclerosis is the main demyelinating disease. It's characterized by a massive immune activation and immune infiltration of peripheral cells into the brain. And these immune and inflammatory cells destroy myelin. And demyelination is the hallmark of the pathology. As you can see here, the disease is characterized by the presence of these active and chronic plaques here in green, which are due to this, as I mentioned, to the destroy of myelin. So there is different phases of the disease, different types, but it is the general characterization, this massive immune and inflammatory activation. So one of the things I wanted to point today is that in addition to these demyelination plaques, we can see in the brain, this is a section of a postmortem section from a patient, the cerebellum. So in addition of these chronic and active plaques, we can see here lesions that have been remyelinated, those called shadow plaques. So myelin is probably one of the few things that can be regenerated in the brain, okay? So this response, this remyelination process works well in the initial phases of the disease, but unfortunately, the age and the progression of the disease decline this capacity. So trying to understand the mechanisms of remyelination and to push this endogenous capacity is a topic in the field and in which we are working. Okay, so in general, this response to remyelination can reverse symptoms obviously associated to demyelination. Okay, so remyelination, this response is obviously dependent on OPC, those cells called oligodendrocyte progenitor cells. Those cells have to migrate into the lesions. They have to differentiate into mature and myelinating oligodendrocytes, which are the cells responsible to remyelinated reactions. Okay, but in addition of these cells, the OPCs, there are other cells which play a key role in this response, which are microglia macrophages. Okay, so these microglia macrophages migrate into the lesions even earlier, and they are essential because they have to clean this excess of myelin debris in the lesions. Okay, so these debris, the myelin debris in the lesions, block oligodendrocyte differentiation and therefore remyelination. So in this sense, the efficiency of microglia and macrophages to remove and to metabolize also myelin is going to be essential for remyelination. And therefore, these cells could be a target to promote or boost the remyelination capacity. And in the last years, we have been talking in this sense, in this topic, and trying to find targets to boost the microglia capacity to remove myelin and to metabolize myelin. Okay, and one of the targets we found was the purinergic receptor P2Y4, which I don't need to introduce in this audience. It's an ionotropy receptor, and we initiated to work years ago in this receptor because we observed that the receptor was highly upregulated in samples from MSA patients, but also in the animal model. Okay, so the animal model we use in general, which is the most accepted model of the pathology, is a model based on the immune reactions. It's called EAE, or encephalitis autoimmune experimental. So what we did to induce this model is to immunize the mice with myelin proteins. In particular, we are using this myelin oligodendrocyte glycoprotein. We inject the mice with adjuvants to boost the immune reaction into the mice. And the mice develop a massive demyelination, particularly in the spinal cord. And this demyelination is obviously characterized by severe symptoms, neurological symptoms, particularly motor symptoms, which we can follow as you can see here in this scale. Okay, the mice start to have symptoms around 10 days post-immunization, develop a peak around 15 to 17 days, and then a partial recovery. Okay, so we started to check years ago what was the role of this receptor, which was highly upregulated in the pathology, P2Y4. And we use the antagonist, which is not very selective, the TMP ATP antagonist, to block the receptor, and surprisingly, the blockage of the receptor exacerbated the pathology, as you can see in the graphs. Okay, whereas in contrast, treating the mice with the allosteric modulator ivermectin increased or ameliorated the pathology. As you know, ivermectin is an allosteric modulator of the receptor, which increases currents mediated by the receptor, that you can see here in the patch clamp recordings. Okay, so we perform a full characterization of the impact of this receptor in microglial cells, and we know that this data has already been published, that ivermectin modulates microglia activation. In the presence of ivermectin, there was an increase in the release of anti-inflammatory mediators and also factors like BDNF, which could increase the myelination capacity of oligodendrocytes. But we also detect that P2Y4 controls myelin phagocytosis. Okay, so briefly in those experiments we performed already, we incubate, those are in vitro, we incubate the cells, microglial cells, with myelin debris, and we check two things: how the cells take up myelin, the endocytosis response, and how these cells metabolize myelin thereafter, 24 hours later. And in general, what we detected was that in the presence of ivermectin, there was an increase in both myelin endocytosis, the endocytosis response, but more importantly, there was an increase in myelin degradation, which was relevant. And this is probably not surprising for most of you, because you know that P2Y4 is also described to be located in the lysosomes and to control the function of the lysosomes. And here in our cells, in microglial cells, we also observed that the incubation with ivermectin induced an increase in the size of the lysosomes, probably due to the fusion of the endosomes with lysosomes. And more important, the pH of the lysosomes was reduced in the presence of ivermectin, suggesting an activation of the lysosomes, which could facilitate myelin degradation. Okay? So we are still interested in understanding more in detail the mechanism of protection of ivermectin. So one of the things we wanted after this, we finished this previous project, was to corroborate that the targets of ivermectin were first P2X4 receptor and the second microglial cells. So to corroborate that P2X4 was target, we were happy to initiate a collaboration with Eric at the University of Bonn. So Eric has developed this transgenic mice, which was an excellent tool for us, in which he removed the intracellular motif responsible for the internalization of the receptor. Those mice are called P2X4 knocking mice in my talk. Okay, so these obviously we check the impact in the function of microglia. We patch clamp the cells, and we indeed observed that currents were much higher when we block the endocytosis of the receptor, meaning that there was more active receptor located in the membrane in microglial cells. Okay, we have performed a whole characterization of the impact of this overexpression in microglial cells. And I'm not going to go to the whole details, but we know that the motility was altered, was an increase in motility in those mice. Lysosome function was also changed. There were small changes too in the lysosome function, but more important, when we perform a transcriptomic analysis of the impact of P2X4 overexpression in microglial cells, we detect that a lot of signaling pathways involved in immune activation and inflammatory pathways were downregulated. And this is important and reinforces the data we already obtained, suggesting that P2X4 controls how microglia responds to insults and probably to damage too. Okay, and how microglia P2X4 probably could control how the immune system is activating inflammatory conditions, which are very relevant for MS pathology. Okay, so after this characterization, we obviously check what was the impact of P2X4 overexpressing in the pathology. Okay, so this constitutive P2X4 overexpression, those are mice full, which is the promoter, is a general promoter. So we have overexpression of P2X4 in most cells, expressing the receptor in the brain. So in this case, we can see that overexpression of P2X4 is protective in the pathology. But here came the first surprise. So this overexpressing, this protective response, it has a gender dimorphism. That's, as you can see here, we only observe protection in female mice and not in male mice. Okay, this protection was also described, characterized. There was lesions were smaller, the axonal damage was smaller too, and microglia activation was reduced. Okay, so next we decided, as mentioned before, we wanted to characterize whether the target was of the therapeutic effect of ivermectin was microglial cells. And the reason to check that is that, as mentioned before, that P2X4 is not only located in microglial cells, it's also expression in neurons. We record oligodendrocytes in vitro, although responses were very small in comparison to microglia, P2Y4 overexpressing oligodendrocytes, so oligodendrocytes coming from P2Y4 knocking mice have higher current, suggesting that in these cells there is also upregulation of receptors. Okay, so we wanted to check whether the effect of increasing concentration, the expression of receptor only in microglial cells. So for that, Eric provided us also with conditional knocking mouse, in which the overexpression of receptor was only in microglial cells under the promoter CD11B. And again, in this case, there was protection, but the protection was only effective in female mice, not in male mice. Okay, so this opens us a big question. Now, why there was only protection in the female mice? Okay, when we checked, we started to characterize responses in both female and male microglia. We didn't observe big changes. So patch clamp reporting, using patch clamp, we did not detect any difference in the currents. The expression of the receptor was the same, so current was exactly the same in male and female. There were small changes in the lysosomes, but very, very small changes between male and female microglia, probably not supporting this big difference observed in the pathology. Okay, so one of the first questions was obvious. Was the effect of ivermectin also dependent on the sex? So we decided to check this question, so analyze this question, and the answer is no. When we treated the mice with ivermectin, as did in the previous article already published, and we took female and male mice, we did not observe gender dimorphism in this effect, in the effect of ivermectin, which makes the problem even more difficult. Okay, so here is what ivermectin does. So ivermectin is allosteric modulator of the receptor. It increase the inward current, the maximal current in response to the agonist, as you can see here, and this effect is similar in both male and female mice or microglia. But ivermectin also block receptor deactivation in the absence of the agonist. Okay, one thing that we characterize as a tau, which is the time involved in receptor deactivation or closing. Okay, so again, in the case of ivermectin, we did not observe difference in receptor deactivation between female and male microglia, but somehow our data is suggesting that the therapeutic effect that we observe with ivermectin is only reproduced in the female knocking mice. So the obvious hypothesis was that the hormones, the sex hormones, could have an influence. And this is the thing we are checking now. Okay, we have performed studies analyzing the effect of estradiol. We didn't observe big changes, but when we analyzed the effect of progesterone on P2Y4 inward currents, we detected that there was a progressive increase in the maximal inward current in the presence of progesterone, but also that receptor deactivation was partially blocked in the presence of progesterone. As you can see here, there was an increase in the tau. Okay, so suggesting, and this is still a hypothesis, that maybe the fact that P2Y4 expression is protective in female mice and not in male mice is because progesterone is also affecting the channel gating properties, and this is not happening in male mice. Okay, so we are collaborating with the chemists in our university too, and they have designed us with new allosteric modulators of the receptor Those series of compounds are called MSK compounds. We are testing them on using patch clamp microglial cells. And we already have compounds that can increase only maximal currents and other compounds that can increase maximal currents and block in parallel receptor deactivation of the receptor. So testing these compounds in near future will help us to determine whether this is the reason for the dimorphism detected in this therapeutic effect on P2Y4 overexpression, and will also help us to understand the mechanism of protection of ivermectin, which probably is relevant for future developments. Okay, so another line I would like to briefly comment in relation to P2Y4 that we are still unpublished, trying to finish this data and publish it soon, is related to this transcription factor called IRF5 or interferon regulatory factor 5. And we started to work with this transcription because this transcription factor binds directly to P2Y4 promoter and activates the expression of the receptor. Okay, so we thought that this was another way to modulate P2Y4 expression, which was very, very relevant to the pathology because polymorphisms in this transcription factor, the 5, IRF5, has been associated to multiple sclerosis. Okay, so we got the full knockout mice and we started to perform experiments using again the EA model. So one of the initial things we detected was that there was a clear delay in EA onset. And this was expected because this transcription factor is able to, it modulates the immune response and probably could have an impact on immune priming, which takes place here in the first, immune priming, I mean in the periphery, would take place here in the first week after immunization. But once the mice start to develop the symptoms, they arrive to the peak at the same peak at the wild-type mice, and in the chronic phase, we observe a clear exacerbation of the pathology, similar to the findings we observed by blocking P2Y4 receptor. And this finding was very, very interesting because this is suggesting that if transcription factor needs to develop the immune reaction and to control the immune function will be necessary for recovery and remyelination, which was a new idea. So we checked this, to check this idea, we moved to another model just in the field, which we call an immune-free model. In this model, we do not induce an immune reaction. In the contrary, what we did is we injected a toxin, a chemical toxin called lisolecithin into the spinal cord. This drug induced local lesions, local demyelinating lesions, which were obviously not associated to symptoms because the lesions were very small and localized. And the model is very well established, the sequence of events, and this model is particularly used to study the remyelination response, not the immune reaction, as mentioned before. So in this case, again, what you can see in the image so here is that in the knockout mice, in IRF5 knockout mice, lesions were again higher. And we also detected that there was, that there was a problem with the recruitment of the cells which are necessary to remyelinate, which are, I mentioned before, the OPCs. OPCs in the wild-type mice, OPCs are labeled with this marker, OLIC2 in green. Here is shown the lesion, so OPCs in the wild-type mice arrive to the lesion and migrate inside the lesions, wherein the knockout mice tends to remain in the periphery, in the rim of the lesions. Okay? In addition to this problem of OPC migration or arrival into the lesions, we also detected that deletions in both cases in the EA model and also after LPC were full of myelin debris, okay? At a time point, with a later time point, which the microglia microphage function it needs to already remove all these debris. Okay, so to analyze whether microglia phagocytosis or metabolism of myelin was altered in this knockout mice, we move back again to in vitro. As shown here, we incubate the cells with myelin labeled in green. Here you can see the endocytic endocytosis 1 hour after adding myelin into the cultures, and how the cells metabolize myelin 24 hours later. As you can see here in the wild-type mice, the cells take up the myelin and metabolize very quickly. There was a massive reduction of myelin in wild-type mice 24 hours later. But in the knockout mice, the endocytosis was partially reduced, but what's more important is that myelin degradation was completely blocked. So in order to check the impact of IRF5 deficiency in microglia, we performed transcriptomic analysis. And again, one of the signalings which was altered was the endocytosis, the clearance of lipids, and in general the homeostasis of lipids. Here, more in detail, where we observed reduction of lipid of genes involved in the endocytosis, genes involved in the release of the metabolites from the lysosomes, and release in cholesterol homeostasis. So as a short introduction, I have to say that myelin has a high content on cholesterol, and free cholesterol is toxic to the cells. So in general, the cells need to, the homeostasis of cholesterol is very well controlled, and cholesterol needs to be removed from the intracellular of the cells by 2 pathways. It's either stored in lipid droplets in the form of esters, or it's released into the extracellular space through specific transporters. So what we detect in the knockout mice was that there was downregulation of genes involved in the transport of cholesterol, and also genes involved in the transcriptomic regulation of all the whole cholesterol homeostasis. Okay, so this was suggesting that probably myelin degradation was altered in this deficiency, in microglia deficiency in IRF5. And to check more in detail this idea, we perform an analysis of a metabolomic analysis of myelin metabolism in the cells. So we incubate the cells, microglia cells in vitro, with higher quantity of myelin for longer time, 24 hours, and then we analyze metabolism by mass spectrometry. And one of the most relevant findings was that there was an increase in cholesterol esters in knockouts for IRF5. Again, suggesting that lipid homeostasis in these cells was altered. Okay, so an alteration of lipid or lipid homeostasis in microglial cells could lead to an aberrant accumulation of lipid droplets, and indeed we went back to the lesions, to LPC and also to EAE lesions, and we detected this accumulation of lipid droplets stained with all red in the knockout mice, both as mentioned before in LPC, but also in EAE. And we also observed this in the lesions. Here, we also observed this accumulation of cholesterol crystals, which will be relevant because it has been described that accumulation of these both lipid droplets or cholesterol crystals in microglia and macrophages could activate inflammasome and exacerbate the inflammatory immune reaction. So finally, to check whether this the exacerbated damage observed in IRF5 knockout mice was due to this mechanism, we treated the mice with two drugs. One was cyclodestrine, which increased the export of cholesterol, and the other drug is an agonist of the nuclear receptor, the RX, which is called GY3965, and these both in general increase the export of cholesterol, and so here both drugs reverse these exacerbated symptoms and recover the remyelination response, and in both cases we detected that the lipid droplets accumulation was reduced. Okay, so as a summary, so we have described that P2Y4 controls microglia activation, but also microglia phagocytosis of myelin, and this response is essential to promote an efficient remyelination. The new data is that the transcription factor IRF5, which controls P2Y4 expression, modulates the lipid homeostasis and cholesterol homeostasis, and has also an essential role in remyelination. So finally, I'd like to thank all the people involved in this work. The PhD student, I already mentioned them along the talk, but also particularly like to thanks our collaborators, Eric, the people working in lipids, which has helped us, and all our supporters. Thanks a lot.
Thanks a lot, Maria, for this nice talk. A bit long, but really nice. And
sorry.
Okay, it's okay. So I will let now David to introduce Ken for the last talk. So I don't know if you can see, I think that there is some problem. Some people are able to see the question in the Q&A window. Check. Now, theoretically, everybody should be able to see it. Otherwise, you will raise your hand at the end, or we will read the question. So, David, when you want. And also, just at the end, we will have also Francesco, who will want to take the to talk a few seconds to announce the European Purine meeting after the talk of the king. David, we don't hear you, David. You have to open the mic. Yes, you are activated now.
It's okay?
Yeah.
Okay. So, hello everyone. Thanks for the nice talk, Alexander and Maria. And I would like to introduce Ken Jacobson, which is Chief of the Molecular Recognition Section in the Laboratory of Bioorganic Chemistry at the National Institute of Diabetes and Digestive and Kidney Disease. It's a very long lab name at the NIH. And as you know, all is a medicinal chemist and is very interesting in the structure of GPCR and particularly the adenosine receptor and nucleotide receptors. And it's a great pleasure to have him here. And he will talk about positive allosteric modulators of the A3 adenosine receptors. So the floor is yours, Ken.
Your microphone is off. Okay.
Okay, thank you very much, Eric and David and Amel, for the invitation. Bonjour à tout le monde. And I'm now going to tell you about recent work with the A3 adenosine receptor. Let's see. Can you all see it? Great. So as you realized we've made a lot of the chemical tools that are used in studying many of the purinergic pathways, particularly the G protein coupled receptors, those being the 8 P2Y subtypes and the 4 adenosine receptor subtypes. And this talk will focus on the A3 receptor, for which we have agonists that have anti-ischemic, anti-inflammatory properties, and 4 of our compounds are actually in clinical trials. Those are nucleoside agonists, but I'm not going to emphasize that in the talk. What I will emphasize are allosteric enhancers. I have no disclosures. 2 of our compounds, the prototypical A3 agonists IB-MECA and chloro-IB-MECA, are already in phase 2 and phase 3 clinical trials for autoimmune inflammatory diseases, psoriasis, and liver conditions. And also the same company is working on an allosteric enhancer, LUF6000, that we have developed in collaboration with Odd Iserman. So initially we were working blindly. That is, we didn't know the 3-dimensional structure of the receptor targets. And now we have that luxury. And we've collaborated with a whole network of Ray Stevens in determining the X-ray structure of multiple purinergic receptors, and we're continuing that process with cryo-EM. But I just want to point out that our determination of the agonist-bound A2A receptor was very critical in understanding the binding not only of A2A agonists, but also A3 agonists by homology. And here is one of our highly selective A3 agonists. So turning now to the positive allosteric modulators, or PAMs. Dr. Gao in our lab started working on this project over 20 years ago now. And he initially took some of the antagonists, adenosine antagonists, based on an A1 antagonist series from Iserman's lab to our lab, and found that unexpectedly, some of the compounds enhanced A3 activity. And then through careful chemical enhancement of that property, we published on LUF6000. Now, LUF6000 only enhances the Emax, the maximal efficacy of agonists at the A3 receptor. It doesn't left shift the activation curve. And if you see in a comparison of various readouts of A3 activation, you see there's possibly some bias towards the cyclic AMP pathway. Recently, we published the analysis of genetic upregulation and downregulation of #1 and A3 agonist, MRS5698, 10,000-fold selective for that receptor subtype, with or without the combination of LUF6000. And for the first time, in an endogenously expressed A3 receptor, we saw a modest efficacy enhancement of the modulator. And we saw that there was modulation of both metabolic and inflammatory pathways. Now turning to the medicinal chemistry that was carried out in our lab by Lucas Falat and Rama Suresh, we have modified the cyclohexyl ring of LUF6000. You see R1 has been increased from 3 carbons to 6, the LUF6000, all the way up to C12. And there's an interesting pattern here. The very small rings are not enhancing. If anything, they're slightly antagonizing the A3 agonist effect. So you go up to an optimum between 6 and 9 carbons in the ring. 9 looks very substantial, but then as you go to larger and larger rings, you lose it. So there is a distinct dependence on the ring size. So what's happening here? It turns out that the analogs of LUF6000 bind to the orthosteric site as well as the allosteric site. So this compound, the cyclopropyl analog, is only orthosteric antagonist, while the LUF 6000 has a major activity as a positive allosteric modulator on the intracellular side of the receptor, and I'll get to that in a moment, but it also has a minor orthosteric antagonism. And as medicinal chemists, we would like to eliminate the antagonism because that's counterproductive if you want a pure enhancer of endogenous adenosine. So we showed by molecular modeling that the cyclopropyl analog does bind very well to the orthosteric site and has the conserved motifs known for orthosteric antagonists. And if we do inhibition of a radio-labeled antagonist for the orthosteric site, we see known antagonists have a clear sigmoidal inhibition curve, but also LUF 6000 and the cyclopropyl analog are not as potent, but they do inhibit the orthosteric binding of this radio-labeled antagonist. And even the open chain analog, MRS 7551, remember the cycloalkyl group is here, and we found out that we can open this ring. And this is a heptyl derivative that seems to be a little less potent at the orthosteric site, and that correlates with the increased allosteric effect of that compound. So now, are the allosteric modulators biased in their activity? There was a hint of that in the earlier slide from Dr. Gao. So first, John Aukenbach and Courtney Fisher at the Medical College of Wisconsin characterized the transducerome of the A3 receptor using nanoBret constructs, and they showed that indeed there's activation of all 4 GI type proteins, and there's also activation of beta-arrestin 2. However, there is even some bias to chloroadenosine, which is so close in structure to adenosine, seems to have some bias in not being able to activate G-alpha O. And interestingly, this is the first indication of bias. So what about the allosteric enhancers? And by the same analysis of adenosine itself. Now we have to keep in mind that PAMs can be probe dependent. That is, they can have different effects on different agonists. So that's why John used adenosine itself. There's no enhancement of the beta-arrestin pathway, but there's a clear enhancement of the G-alpha OA and G-alpha I3 isoforms. And in fact, these isoforms are enriched in immune cells, which we believe is the pathway of a lot of the beneficial effects of A3 receptor activation. So then we went on to examine the structure-activity relationships at this C2 position and the 4 position as well, and also made heteroatom substitutions in the heterocyclic core and the exocyclic NH. So we have a pretty good handle on the SAR, structure-activity relationship, all around the molecule. So do A3 PAMs have any anti-ischemic properties in vivo? Well, John Aukenbach did show that there is a cardioprotective effect of an open ring analog, LUF 6096 from the Iserman lab. He showed that it did work in vivo. However, it had a very short half-life. So our subsequent structure-activity probing did not use this as a lead compound. And interestingly, at mouse A3 receptors, there is only a hint of enhancement by either LUF 6000 or LUF 6096. This is much less than the enhancement at the human receptors. So we used that insight to make chimeric receptors to try to locate the allosteric binding site. There were a number of modeling exercises, including one of our own, that predicted that the extracellular loops are involved in the binding of this PAM, and that turned out to be completely wrong. So we made chimeric receptors where part of the human A3 receptor was replaced with the mouse sequence, that is the non-responsive sequence. So we eliminated the outer side of the A3 receptor as the site for binding of LUF 6000. So that means it's completely separate from any of the orthosteric residues. And then we went on to sequentially block out these other regions by inserting the mouse sequences. So TMs 3 through 6 are not involved, leaving TMs 1 and 2 and TM7 in the final C-terminal helix 8. So it turns out that by substituting the mouse at this position, you get a reduction but not total elimination of the PAM. But if you substitute this region of the receptor, you get total elimination of the PAM effect, which is interesting because there was no known compound binding to this intracellular site on the receptor or at any other GPCR. There's some indication in other receptors, but there's no clear assignment of the binding site. So now we have identified these two tyrosines as potential interacting partners with the allosteric enhancer. This was done purely by modeling. Molecular modeling has gone far. So even though there is no primary experimental structure for the A3 receptor, we've been able to use the AlphaFold prediction and cryo-EM structures of A1 to clearly identify these tyrosines as important. And when Courtney Fisher and John Ockenbach's lab mutated these tyrosines either to cysteine, as in the mouse A3 receptor, or to phenylalanine, which simply removes the hydroxyl group from this tyrosine in helix 8, we get a complete loss of allosteric enhancement. So we think that the final stable posed, predicted by Matteo Pavan in our lab, involves both hydrogen bonding, in one case with TM1, which explains why changing TM1 had a modulatory effect. And also, these two tyrosines are key, both in pi-pi interaction and in hydrogen bonding. So let's see the details. So Matteo, following on the lead of the initial results from Veronica Salmazo in our lab, did molecular dynamics simulation, and he showed which residues are predicted to have stable interactions with the allosteric modulator, and both tyrosines indeed have stable interactions. Here you see in this very long simulation, we see that the position of the modulator is stable. So that means that these two portions of the molecule, the dichlorophenyl moiety and the cyclohexyl moiety, are quite lipid exposed. In this case, it's completely lipid exposed in the phospholipid bilayer. In this case, it's partially in contact with hydrophobic residues of the receptor, but it's still, on the other side, exposed to the phospholipid bilayer. So here's what the actual pose looks like. So you see here on the side view, here's the cyclohexyl ring, and here are the fingers of the hydrophobic residues of the receptor that are creating a relatively small, sterically limited pocket for this PAM. Now that explains why when we made this cyclohexyl ring too large, it exceeded the size of the pocket. And then the dichlorophenyl group is pointing clearly into the phospholipid lipid region. So then the question came up, how do we now continue the medicinal chemical process of optimizing these compounds. And we tried in the past to put hydrophilic groups on these two regions, and it was mostly unsuccessful. Well, now this new pose that's identified explains why hydrophilic groups are generally not tolerated in both of these two regions, because they're penetrating the lipid portion. So this, as we see it, this LUF 6000 has to embed first in the phospholipid bilayer and then find its binding site. And that occurs through a multi-step process according to Matteo's molecular dynamics. First, there's a hook. This NH on the PAM does not like to be completely surrounded by lipids. So it's searching for a hydrogen bond, and it finds that, according to Matteo, in the OH moiety of tyrosine in helix 8. And then other stabilizing interactions form. There's a water-mediated hydrogen bond, and this phenylalanine seems to flip in and out of the ring in the apo form of the receptor. So when it's out, it allows this tricyclic heterocycle to form a stable interaction with both tyrosines inside the binding site, and that establishes what we're calling the stable pose, which you've seen before. So finally, let's see how we've utilized this structural insight to enhance the activity of the PAM. So here you see the phenyl ring. So the two chloros have been removed, and we've attached very hydrophobic chains that are coupled through a C triple bond C. That's because of the chemistry. A Sonogashira reaction makes this a very easily attainable series. So Balaram in our lab did the whole series from very short to very long. And this enhancement, you see there's an enhancement of the efficacy of chloroibumeca. And there's also, for the first time in any of our studies of these PAMs, there's a left shift of the activation curve of chloroibumeca, suggesting an enhanced potency as well. And that whole thing is optimized in the case of 9 methylenes in the chain. And so you see a substantial left shift. There's no indication of orthosteric inhibition of the effect. So this is a substantial qualitative and quantitative enhancement of the effects of these PAMs. So here's how we analyze what's happening with this long chain. And we have a term for this: lipid trolling, like fishing. So we've extended the chain and we are actually accessing the negatively charged phospholipid head groups on the inner leaflet of the phospholipid bilayer. And this is not just a cartoon that we came up with. We started with a cartoon, but then Matteo has actually validated this length dependence and shown that in the case of the 9-methylene analog, it forms a stable interaction with the simulated negatively charged phospholipids in the membrane. So we have very good support for this, both in the SAR, the pharmacology, and the molecular modeling, and we really love that situation. So now, in summary, we've started with empirical probing, finding the series of imidazoquinolinamines just by chance happen to have a secondary action as A3 receptor PAMs. And we have then used site-directed immunogenesis and molecular modeling to identify a site. And it will be interesting to see if cryo-EM studies can actually confirm experimentally this site that we have so much support for from the other experiments. And by extending the hydrophobic chains from the 4-phenylamino position, we can actually access the phospholipid head groups of the inner membrane leaflet. And we think this could be a model for new type of modification of modulators of GPCRs that can extend into the phospholipid portion. This is a new mode for us, and maybe we can lead the way in the terms of this modeling. So I thank the other members of our group, most of whom I've mentioned along the way. This is actually including people who have left our group. Our group is only about half this size. And I thank you for your attention.
Thank you very much, Ken, for this very nice talk. And thanks for coming, all the speakers. Before we go for the question, maybe Francesco can, I don't see him, but can you, okay, hi, Francesco. It's your time to.
Hello, good afternoon to everybody. If I may, I would like to show a slide.
You can share something if you want.
Yes.
Yeah, you can.
Yes. I'm going to share it. Do you see it?
Yes.
Yep.
Yes.
Yes. Okay. This is just an advertisement for the upcoming European meeting, which I'm sure you all are well aware of. And just to remind you that we are going to meet again in a few months in Ferrara for the second European meeting, which is a follow-up of the first European meeting that was organized by the late Teresa Miras, Portugal, in Santiago de Compostela 3 years ago. I'm sure that this will be a very important occasion to me to discuss science, to discuss both P2, P1 receptors, ectonucleotidases and transporters, and so on. And the meeting will be organized with plenary talks, with lectures, and with a round table. And the round table will be, in my opinion, very exciting because it will analyze the main reasons why clinical trials, anti-cancer clinical trials with drugs and antibodies targeting the purinergic pathways have not so far provided the results that we all hoped for. So I think that this will be a very important issue for discussion. And this is it. I hope to see you all in Ferrara within a few months. And thanks again to David, to Eric, for giving me the possibility to advertise the meeting. Thank you.
Thank you.
Thank you, Francesco. Of course, we will be all there. Will be cool to see everyone. That we have few time for the questions. So there are some questions in the chat. So I pick some. There was one question that maybe you answer, Alexander. What is the effect of coffee consumption of brown adipose tissue? And does it increase obesity?
Yeah, I typed it already. This is why I was always typing
okay.
other talks. Yeah, so that's for sure an interesting question. But since caffeine is not only inhibiting adenosine receptors, but also phosphodiesterases, it's very hard to, how should I say, to figure or to just predict it. So I ask again my co-workers, hopefully they will put some caffeine on these adipocytes soon so that we can finally answer this question. It's a good question, thanks.
If I may add something, if you provide caffeine through drinking water to mice fed with high-fat diets, you find an improvement. We have done some experiment a long time ago with that, and you protect from high-fat diets when you provide caffeine through drinking water. So, but I agree, maybe it's not an adenosine receptor-based effect, could be something else. But
and there could also be central effects because it's centrally regulated for sure, the brown
of
fat.
course, but we can discuss that when you come.
I just have to find people that do the work because you don't want to know.
I had a question also to you, Alexander. It's what do you think there is a link between hypothalamus through the vagus nerve to the B18 innervation by the vagus nerve, and the A2A can or inosine could play a link between the brain and the B18 connection?
Yeah, I already replied to you also via, I also gave you the citation. There was a paper, recent paper about secretin. So there is a hormonal link also between hypothalamus, fat, and the intestine. So it's very complicated, and you are the neuroscientist and much more clever than I am to figure that out. But
that's not true.
There appear to be different hormones and things.
Yeah, Rodrigo is laughing because he knows that the neurosciences are not that clever.
No. Here in Bonn, it's most important, and I'm just a simple fat person, so I.
In the audience, if you have questions, please raise your hand, your virtual hand, please. There is a question from Valerie to Maria. As the new positive allosteric modulator of P2X4 has been tested on P2X7, and what are the selectivity?
Sorry, yeah. So no, we haven't tested it yet. So the conditions where we are using to characterize the effect of the allosteric modulators on P2X4 are low concentration of ATP. So we are not activating on those conditions P2X4 receptors. So the answer is we don't know.
Okay. Another question from Rodrigo to you. Is the number of microglia altered upon knocking in P2X4 receptors?
It is definitely. So in the experiments of EAE, there was a massive increase in the number of microglial cells, in massive microgliosis. And in the presence of the antagonist, TMP, there was a further increase in microglial cells in the MS model. Okay.
Okay. A question from Rodrigo to Ken. Is there any evidence of A3 receptor partners that may hinder the possibility of binding of A3 receptor PAM?
No evidence that I'm aware of. No.
We don't hear you, Rodrigo. You can put your mic on.
I was just thanking Ken for the answer. Sorry.
Let me check. Yeah, I had a question to Alexander about the possibilities that inosine released by microbiota from bacteria could eventually act on the BAT.
I think this is a very important question. I already wrote that to you, and then I owe you a beer because
okay.
I did not yet look at it, and I was just starting to look at it. I think it's very interesting.
Okay, I have to send a paper. There was a paper some years ago about.
Yeah, I just saw it. It was in Science. As I said, I owe you a beer.
Okay. Oh, only one. Not enough.
Okay, two. One in Ferrara, one in Lille.
Okay. To Maria also. Sorry.
I know. Go.
Is IDF5 control P2X receptor expression in neurons?
Okay, so there is no data or literature about that. So the only paper I already mentioned is on microglial cells controlling P2X4 expression after pain. And our data is also only on microglial cells, so have no answer. But I presume the answer is no, because this is a transcription factor involved in immune response, and I think neurons do not express IL-5.
Okay. We have I have a question for Ken. Do you see some application of the new PAM for A3 in neurological disorders, or eventually neuropathic pain? You think that it can be
Yes,
of interest?
definitely. We're very interested in applying it to neuropathic pain. And in the past, we've been limited by the relative inactivity of the PAMs like LUF 6000 in rodent species. So we couldn't put it to the test effectively. But now with the new long chain positively charged analogs, we might have an opportunity to do that in experimental animal models. And as far as getting into the brain, we have to work on the formulation of these compounds because they have the positive charge, but yet on the other end, they have a very hydrophobic moiety. So I don't know, we haven't studied the pharmacokinetics of those compounds yet. So that remains to be seen.
Okay, thank you. I have another question from, okay, so Alexander answered to Herbert about other effects of the loss of nucleoside transporter on the patient. So you answered. I have a question, another question for Alexander. Is the SLAC29A mutant loss of function also associated with lipodystrophy or only underweight?
Yeah, so lipodystrophy, they didn't look, and I'm not aware of it, or that they have lipodystrophy, but the patients or the people that have the mutation tend to have lower body mass and lower body weight.
Okay.
And their chances to get obesity are much smaller.
Jean-Sébastien had many other questions, but I think you can ask them when you will be in Lille with a beer. Sorry, I check in. You see other question, Eric, in the chat.
There is many already answered.
Yeah.
Sorry, I'm a bit lost in the chat.
Many questions.
There was one also to Alex about the irisine, but you.
I tried to answer.
Okay, you try to answer. Okay.
Yeah, but if there are, I mean, people could still say or ask their question live, isn't it, if there is something urgent?
There is also a question from Rodrigo to Alexander. First, he congrats you. There is a question about the A2A increasing norepinephrine or acting on adipocytes. There seems to be a time shift between norepinephrine versus ATP release. Is it true? Rodrigo, maybe you can ask.
Yeah, so I was looking at it again, so I don't think that there's a time shift, but the concentrations are for sure not the same. I think that is a very fast turnover then of the purines, and so at least in this ex vivo stimulation, we see a clear increase. I can show perhaps again. Yeah, anyway, I mean, there's not a big lag. I mean, it's I think this is mostly released from the, it could be either released from the, wait, I show again. I get to your point. I mean, the question is, I think here, is it direct release with the vesicles that contain the noradrenaline? So here, this is very fast. Or is this lag here 2 minutes, 3 minutes perhaps? Here we have the peak at 4 or 5 minutes. Here it's already at 3 minutes. Whether this is due to release of the purines by the adipocytes and not by the nerve endings. I think that's a very good point. I mean, yeah, we are trying to figure that out in the when we put noradrenaline on the adipocytes per se that don't have any vesicles anymore, or with vesicles, no nerve endings, then they can release ATP and adenosine themselves. They do that constantly, so to say, also. It's like a cloud of purines around them.
Rodrigo, you want to add something? You have
It's just trying to refine the question that I had in mind to Alexander. First of all, congrats for the very beautiful story. Could it be that the effects operated by adenosine are due to an induced release of noradrenaline instead of a direct effect on the adipocyte?
Yeah, so it's like this. So if you have no, so if you just have the pure brown adipocyte cell culture, and if you put A to A agonists on them, or also A to B is also working, they are nicely activated. So they don't need the norepinephrine. But what I think is very interesting is that it appears to us that norepinephrine is the shot, so to say, or the signal for the cells to be activated. And then if you think about it, I mean, previously our ancestors or the mice that are in the woods or the mammals, they are exposed, at least in Germany, for 4 months to cold temperature. So if they have an E all the time, I think that's not sustainable. So I think they need the purines, this cloud of purines around them, to keep a sustained activation of this thermogenesis, or they would freeze, I mean, immediately to death. So I think it's a synergistic mechanism.
Thank you. One question from François to Maria. Given the involvement of P2X4 in neuropathic pain, do you think that ivermectin would be a real potential therapeutic strategy in multiple sclerosis?
Yeah, that's a very, very good question. Okay, so it's true that multiple sclerosis could have neuropathic pain, but it's not the most important side or symptom in the pathology. So when in the EA model, we tested the impact of ivermectin in pain induced by in the EA model, and we did not show any exacerbation of the pain. Okay, but I don't know if this will be translated or has an impact in patients.
Okay, thank you. I have a question for Ken, Alexander, and Maria about the A3 PAM potential in metabolic disorders and in multiple sclerosis. Do you think that the A3 targeting with your new compound could have some, let's say, a future in this area, Ken? Because Alex told about the adenosine and inosine in BAT, but what about the A3 in this mechanism? And the same question for multiple sclerosis, notably regarding the inflammatory part of multiple sclerosis.
Yes, I think there could be potential. Certainly, we know that A3 reduces neuroinflammation. This was reported recently, most recently by Daniela Salvemini, our collaborator at St. Louis University. Also in the liver, the A3 receptor has some benefit in animal models of obesity. So there could be application of these PAMs in both neurodegenerative and metabolic disorders. And as we know, the PAM in principle could be silent until there's a local elevation of adenosine. So that could be an advantage over a pure A3 agonist.
Okay. And we know if brown adipose tissue express the A3 receptor?
Yeah, I'm just checking. So we always compared brown and white. And so we found that, for example, the A2A receptor, at least in mice, and as far as we know, also in humans, is very highly expressed in the brown and not so much in the white fat. But the A1 receptor, the inhibitory for sure, GI coupled, is highly expressed in the white and not so much in the brown. And also the A3 receptor is very highly expressed in the white fat versus brown fat. And we also use then the different agonists. And I'm just checking, I think the A3, there was not a lot of effect there. I mean, at least the cells that we checked now, but I mean, that would be something for sure. In theory, if you would then block an A3 receptor, then you could perhaps increase more energy expenditure, or use an A2A agonist. I think that would perhaps be possible. But from the data that I just was quickly looking at, there was not a lot of A3 receptors there, at least in the cells we looked. Thanks. Very good point.
Thank you very much. So I think we are done with the question in the chat, right, Eric?
I think so, yeah.
Yeah.
So is there any other question with the people there? Rodrigo, because your hand is always raised.
Sorry, took it down. Sorry.
Okay.
Yeah.
So I think we are done for this session, and we